Hi everyone, this video is about eukaryotic transcription initiation by RNA polymerase I pre-initiation complex. RNA polymerase I transcribes ribosomal RNAs. Ribosomal RNAs, along with ribosomal proteins, are components of the ribosome. I will start by talking about the ribosome. Ribosomes are macromolecular machines found within all living cells that perform biological protein synthesis. Thus, the quantity of ribosomes substantially affect all biological activities. Here is a crystal structure of the eukaryotic ribosome, showing how it's made up of a 40S small subunit, illustrated in blue, and the large 60S subunit, shown in yellow. In the structure, proteins marked with dark blue and yellow colors are located predominantly on the periphery of the ribosome, whereas rRNAs, marked with light blue and yellow colors, are located predominantly in the interior of the ribosome, with the rRNAs playing catalytic functions in the ribosome. The synthesis of rRNAs is an essential step in ribosome biogenesis. The levels of transcribed rRNA strongly affect the total amount of ribosome produced. In eukaryotes, a ribosome contains 4 structural ribosomal RNAs, 5S, 5.8S, 18S, and 28S rRNAs. The 5S rRNA is transcribed by RNA polymerase III, which won't be covered in this video, whereas the other 3, 5.8S, 18S, and 28S, are transcribed by RNA polymerase I from a single promoter as a polycistronic transcript that then gets processed into individual rRNAs, as shown in this schematic. There are several mechanisms that control rRNA transcript levels, such as rRNA gene copy numbers in the cell and the efficiency of the pre-initiation complex formation for RNA polymerase I on the rRNA gene promoter, which will be the focus of this video. So, RNA polymerase I and its pre-initiation complex is responsible for the synthesis of these 3 structural ribosomal RNAs. Let's see how this RNA polymerase I pre-initiation complex is assembled. First, the multimeric promoter selectivity factor, abbreviated SL1, binds to the core promoter of the rRNA gene. SL1 consists of a tata-binding protein, TBP, that works with 3 or more TBP-associated factors, abbreviated TAF1s. Then, a homodimeric DNA-binding protein, meaning that it's comprised of 2 identical polypeptide chains, abbreviated UBF, binds to an upstream element, abbreviated UCE. Then, SL1 recruits polymerase I through TIF1A, acting like a bridge. UBF stabilizes the binding of SL1 to polymerase I at the rDNA promoter. After the completion of the pre-initiation complex, polymerase I is cleared from the promoter and moves on to transcribe the rRNA gene, regulated by TIF1A. RNA polymerase I continues to transcribe until it reaches a termination element that leads to RNA polymerase I dissociation. Finally, pre-rRNA is processed to structured rRNA, 18S, 5.8S, and 28S rRNAs, which become incorporated into the ribosomes along with ribosomal proteins. Thank you for watching.